if the peptide is over 50 amino acids, okay, then what we have to do is isolate that protein. Remember, we spend a lot of time isolating a protein. We have a big sample. We take a small amount of that sample and we break it up into small chunks. Okay? And if we can break it up into small chunks, then each chunk can be sequenced. Okay? Oh, well, man, now I got 20 chunks that are sequenced, but I need to know my 1,000 amino acid sequence. What goes first? Okay, that's a little bit harder to do. I've got individual pieces, but once you break the peptide, I've lost that information of what the sequence is. All right, so what do? Well, remember, this is not search, this is research, so we start over. We go back to our purified protein, we take out a small amount, and we cut it up, but this time we cut it in different places. Okay, we sequence all of those. And what you're going to do is get a whole bunch of data. All right, now, how do we fragment the peptides? We can actually do it chemically or enzymatically. Chemically is a little bit older. There are a couple chemical pathways to break. But what you want to do is you want to break every single protein in your sample in the same places. Okay? So that you can purify the fragments, and all the fragments are the same size. Chemically, we can do that a couple ways. Most of the time, we use enzymatic methods. What we are going to use is an enzyme whose whole job is to break proteins. Okay? And they tend to do it this way. They will only break at certain places in the protein. And so the enzyme trypsin will cleave on every carboxy side of a positive residue, arginines and lysines in this case. So anytime there's an arginine and lysine, it's going to cut it. And it'll cut every protein in your sample. And all the fragments will be the same, will be similar in size. Then you separate them off by size, and you have different bands. You might break it up into 20 different fragments, but you have to have each of those purified fragments the same, and you sequence them all up. Then we can use usually a different enzymatic. And so we usually use enzymes. Trypsin, chymotrypsin, protease, there's a whole bunch of them that we can use. Which ones do we use? Well, which ones are in your refrigerator, or which ones you order from the company. And so sometimes you have to use different ones because you might fragment it larger than 50 using certain ones, and so if they're above 50, this doesn't help you. Once you have two separate sets of data, one, all the sequencing from one cleavage point and all the sequencing from a second cleavage point. Then what you can do is you can use overlap. Now this is a wonderful technique for computers to do, although way back before computers you had to do this manually. And you can see a chymotrypsin peptide has valine, lysine, alanine, tryptophan. Okay, that's great. But when I compare that to another enzyme that breaks peptides in a different way, you can see the overlap. And so the val lys at the end of one peptide and the ala-ala-trip will match up and overlap, and you get a longer peptide. And you continuously work that all the way down. Can be done manually. Really, the computer aligns this really quickly. It's a very easy computerized item. Okay, and you can get the full sequence of things that are longer than 50. All right, so we can take and purify a protein. We can get its amino acid composition. We can then get its sequence, and we are starting to know a lot about the protein. All of which is, up to this point, what level of protein do we know? Primary. There's secondary, tertiary, and quaternary still left. So we still got a lot of work. Okay? One thing that we can do that's kind of easy to do, and it intersects sequencing, is to try to find disulfide positions. Okay? And so you can take your sample, again, you go and you have your very precious sample that you've been working on. You take a small little part out, and you can break it up like you're doing sequencing. But most of the time when you're breaking things apart, you also use beta-mercaptoethanol to make sure you break the sulfides because you want to get everything unraveled. This time, what we're going to do is we're going to break the protein without breaking disulfides. Okay? And so you break the peptide bonds, and you get peptides. Okay? But what you're going to get, if there is a disulfide present, you're going to get one little peptide that are connected covalently through a disulfide. They will act as if they are covalently linked together because they still are covalently linked together. And you will do electrophoresis by size. Okay? And so you'll separate by size, and you get this banding, remember, as you get separation. And then what you're going to do is something really odd. You're going to treat the gel with performic acid vapors. What performic acid will do is it will break and oxidize the cysteines. All right? Then what you do is you take that gel that's separated this way, you take it and you flip it on its side. Okay? And you run the electrophoresis again under the same conditions. And so wherever that peptide went, it'll start moving again. If nothing has happened to the peptide, if it moved a little ways one direction, it'll move a little ways in the opposite direction, or the 90-degree direction. If it moved a lot in one direction, it'll move a lot again in the same gel. And what you will do is you will have a whole bunch of marks or a whole bunch of bands that are diagonal. If they are diagonal, that meant nothing happened to them. Nothing changed between the two situations. If it ran a little before and it ran a little again, nothing happened to it. But anything that is off the diagonal, okay, meant that in one direction it was big, connected together. In the second direction, it was broken, two smaller pieces. There was a disulfide there. You can cut them out of the gel, sequence them, and where the cysteine is most likely where the disulfides are. And usually you have one, you might have two, and then you would have to do a little bit more fine-tuning on that. But it allows us to find disulfide bonds. All right, another thing that we can do with peptides. We can break them, but we can make them. Okay, and so we're going to do some synthesis. All right, and we're going to do laboratory synthesis using organic reaction schemes. Now here's the difference. In the synthesis of peptides normally, using the ribosome, messenger RNA, and all of the mechanism that is natural, when you synthesize proteins, you start with the N-terminus and you go towards the C. Okay? When we do it using our chemistry, we do it backwards. We synthesize the C-terminal first, then we add towards the N-terminus. So, our chemistry is totally different than the biochemistry that happens in the body. Okay? To do this, we're going to take a peptide and we have to protect it and activate it in a certain scheme so that we can have the synthesis go in the way that we want to go. All right, our activating species is DCC, dicyclohexyl carbonate. You can read it, all right? It's too early on Monday morning. All right, so what we've done is we have added a leaving group to the C-terminus, okay? This is activating it, meaning that I have made this carbon, okay, the carbonyl carbon, this one right here, active. So if something attacks it, this will leave very good. Okay. I then can add FMOC to the N-terminus. FMOC is a protecting group, making the nitrogen inactive. The nitrogen can be a nucleophile, okay, and attack carbonyls, but if I put this FMOC, it's going to block the ability of this nitrogen to attack a carbonyl. So this amino acid right here is activated on the C-terminus and protected on the N-terminus. No reaction, wants to react a whole lot. Okay? If you put this on in a different order, or we'll say, which one do you want to put on first? The activator or the protector?
Or the protector.
The protector. You have to protect it first, because if you just put on the activator, then what will happen is You put on the activator on one, and now they're all floating around active. If this is not protected, what will happen is you will just make one long chain of one amino acid. Okay? So you have to protect it before you activate it. All right, let's do the start. You know what the sequence is. So what you do is you start with the first amino acid, and you have nothing on the C-terminus and protected on the N-terminus. And you react it with a polystyrene bead. Okay, so one of those pieces of sand. And the sand is activated by having this chlorine here. So what happens is the oxygen attacks at this carbon, the chlorine falls off. Okay? What this does is it makes it a solid state synthesis. What does that mean? Your growing chain is going to be on something solid. So what I can do is have a reaction vessel that has a stopcock at the bottom. I drip in my reagent, let it shake and mix for a little while under a certain temperature, and then open the stopcock, let all the reagents drain out. Then you can wash it, and your growing synthesis will happen on this polystyrene bead, or the sand, and it just enhances separation, makes it more quick. It also makes it automated. All right. And so I've got my first amino acid growing. That makes a very short peptide. Okay. Protected. And now this is not active because it's already reacted, and this is just there to hold on to it. Second. All right. What I do in the main reaction chamber is I take off the FMOC. I do that by adding mild organic base. Okay. So a weak organic base will take off the FMOC. What you have now is a reactive species. This wants to attack things. Okay. I then, in a separate container, will protect the second amino acid with an FMOC, add DCC to the C-terminus to make this active, and then I drip this particular molecule into the chamber that has your reactive nitrogen, and that nitrogen will attack at the carbonyl, kick up, kick down. This goes out, and I've made a peptide bond between one amino acid and the second amino acid. Okay? Very good. All right, at this point in time, I have a very tiny little peptide, all of 2 amino acids long. All right, usually 2 is not long enough. If you're going to spend time, you're going to make something longer. So what you do is you go back up, and now it comes back here. Now I have 2. I add the third. I do the same. I now have 3. Add the fourth, and the same. And I keep that going until I synthesize however long that I want to create that peptide. Okay? When I'm done, the last step is you add hydrofluoric acid. Hydrofluoric acid will do 2 things. It will take off the FMOC protecting group, and it will catalyze the reaction to displace your peptide off of the bead. So it cuts the C-terminal connection to the bead, and it deprotects, and now you have a free-floating amino acid that floats down and out of the reaction tube. Now, the reactions aren't 100%, so it has to be purified a little bit, usually by size, or could be by charge, and you purify that and you get your little peptide that you want. All right. This is the old textbook's picture. I like the old textbook picture for one thing. The way it organized it was a little bit different and actually a little bit more logical. There is one thing, though, in the new textbook's picture that's important. Okay? We had to protect this N-terminus, right? That one, and we had to make it either not reactive or reactive under certain conditions to control where it goes, right? And so we also control the reactivity here, right? So that we make only 2 things active. The N-terminus when we want it, and the certain C-terminus when we wanted it. So those 2 only react. All Are these the only reacting species in this mix? Is this the only nitrogen that is free in every peptide?
Depends on the section.
What you have to watch out for is the R groups. The R groups have places that have, like lysine. Lysine would have a nitrogen that is free that would compete with the N-terminus if you just left it there. And this is where the new textbook does indicate that all R groups have to be protected. There are R groups that have a C-terminus, that have an acid that could have been reacted with a DCC and then could react in inappropriate ways. And so every R group must also be protected. Now, here is the saving grace. Okay? This incoming amino acid that is protected and activated can be purchased directly from the company that are already activated, already protected, and the R groups are protected already. And so when you do this, I actually worked next to a core lab that did this. What you do is you get these little vials and you fill up which amino acid you want and you put it on the rack. And you fill up the next one, you put it on the rack, and you fill up the next one, you put it on the rack, and then you just get a rack of all the amino acids you want. And then the instrument will go in, take out the solution, do the reaction, go to the next one, and it will kick out the little thing, each of those little, sample tubes, and it will synthesize it all nice and neat for you. And so you can buy all of these prepared, and you don't have to do a lot of the protecting and the deprotecting and activating because we can purchase them now. Okay? All right. Once we have that, remember, purified protein. We took out samples to do sequencing. We still have our protein, and you take out another sample. All right? This is what you do. If you spend a lot of time, you always have your main sample, and it's frozen, right? And you actually take that main sample and you freeze it into lots of aliquots of your purified so that you don't even do the freeze-thaw cycle very often. You take out one little. You never have your entire experiment out at one time because if you do, you could lose it all at one time. Not a good thing. When I was doing my research, I was using photoactive material. Okay, so I was synthesizing drugs that would be reactive if you turn light on, certain wavelengths of light. We actually disconnected the light switch in our lab because if you're doing your synthesis and someone just comes in and flips on the light, boom, a month's worth of work all gone because someone turned on the light that they weren't supposed to at the wrong time. And so that is, you never have your entire thing out at one time, or you do it once, and then you have to do a whole lot of work to catch back up. All right. We take out a small sample and we're going to do some analysis. We're going to look at some secondary structure and some tertiary structure. What are these you already know about? Circular dichroism. Using polarized light, we talked about this. So polarized light shined into the sample, and it is proteins are optically active because of really the alpha helix dominating, but then all the other amino acids are also L isomers. That's slightly part of it. Here is the full scope of a circular dichroism. And what the signaling would be. And so you have some dips in certain areas and a peak in certain areas. Now, when you did the experiment and when you did the exam, when you looked at it, did you even see this peak here? Did we actually look at a peak? No. What the article looked at was this region right here, which is looked at much more often. And so the double dip is what we were looking for versus no dip. And so you can look at any place on here, but most people look at that particular signal as the amount of alpha helix. X-ray crystallography is one way to take a picture of a protein. Okay, so why can't you take a protein and just put it in our microscope and look and see the structure? Right? Well, most microscopes aren't that powerful, right? But you can get larger and larger optical, 1,000, 10,000. Why can't we just take a picture of it?
Be affected by the light.
Ah, here's the deal. For you to take a picture of something, the light has to be shined towards it and it has to reflect back off. Okay? Here's the problem with a protein. The wavelength of light, the actual size of the wavelength, is bigger than the protein itself. So it can't hit it and reflect back because the wavelength's too long. So I have to get something that has a wavelength that is on the order of the things we want to hit and reflect back. That's where we have to go down to X-rays. X-rays are in the size range that atoms are. Okay? And so we can't take a physical picture of it because we don't see X-ray. If you do, that's really weird. Okay? And so we just have to use the right wavelength. And so in terms of bond distances, we talk in angstroms. Visible light is about 4,000. Even the smallest visible light, that's just way too big. The X-ray is on the 1.5 angstrom range. All right. Okay, secondly, if I want to take a picture of something, it needs to be still. If something's running or moving, you get a blurred picture. And if I'm trying to get a picture of everything, if they're all moving in different directions, then you just get a mess. So what you have to do is have every protein lined up in the same orientation, still. Okay? That's not what happens in the body. So what we're going to do is we're going to create a salt crystal of your protein. Just like sodium chloride lines up in a regular repeating lattice, we have to force the proteins into a regular repeating lattice. All right, let me tell you this. Crystallizing a protein who is globular in nature, wants to float around in water, and wants to be free and moving, salting them out and making a crystal is a hard job. There are protocols. This is what you do. You add this, you do this, you do that. It's more a art than it is a science, because certain things you just need to have the feel of how it will do. And sometimes you don't get a crystal and you put the sample up in the refrigerator and then 4 weeks later you look at it, oh, there's a crystal. How did I get that? I have no idea. Let's go take a picture of it. And they get a published paper out of it, okay? And so it's kind of hard to get a crystal. Also, you have just now made a crystal. You have made them into a lattice that is not in solution, that is much more concentrated than it was in normal, so the picture you may have is a crystal structure, very detailed, but very out of context of what it really does. So, wonderful bits of information. When we get a crystal structure, it really drives what we do, but there's always a sense of question, is that really the real structure? And so, a crystal structure, even though you can get high resolution and a beautiful picture, it has to line up with all the other experimental evidence for you to believe it. And so, if we put the protein in solution, I always don't know if this animation, okay, good. The animation is actually going. All right, use a salting out agent and, automatically, beautifully creates a protein crystal. All right, never is it that easy, okay? But once you get the crystal, you can put it in a sample tube, and then you hit it with X-rays. Now, what X-rays will do is they will approach the protein, and every time it hits an atom, it will deflect. The larger the atom, the larger the deflection, and how many things are deflected. We normally start to see around carbon. Anything smaller, we don't really see, so we don't see the hydrogens. Okay? But metal centers, the larger they are, the more we see, the better we see. Now, we hit the protein at different levels. And what we do is we get based upon the math, where this deflection was, from where the beam was, there must have been something here. And what we get is what's called a density map. Okay? There must have been something here because the X-ray got deflected over here. We did a whole lot of math. Now, what we do is we move the X-ray and we take slices as we go down the whole protein crystal. Okay? And what we do is we get flat images, and then we take all of those images and combine them together to show where all the densities are. Now the density will give you something like this. Oops. Will give you, okay, there is atoms here. Then what we do is we calculate, okay, based upon this shape, what amino acid fits within that shape? And then we get down to what the actual proteins are, and we see where the carbons and the nitrogens and the oxygens are, and based upon that and knowing Vesper rules, we can put all where the hydrogens are, and we calculate what that particular shape is, and you get the entire protein. You both get secondary and tertiary structure with a crystal structure. Okay. Scattering is dependent upon molecular weight. It's all a mathematical calculation. Really, really complex mathematics. If you like math and you like biology, here's your place to go. You get the backbone. You get good resolution, but it may not be the real structure. Okay? So a protein that I had, there's a wonderful crystal structure of it. The protein is called chemogulin. And chemogulin kind of looks like this. It's a little tiny molecule. It's got a pocket up here and a pocket down there. And they called it a dog bone crystal structure because it looked like this. And so we knew exactly what the protein looked like, right? But here's the problem. Under experimental conditions, the amino acid right up here at the top and the amino acid right down there at the bottom could be linked by a molecule that was this big. This crystal structure had them as far apart as they could ever be. Okay? This was the crystal structure in the crystal. When it is in solution, what happens is it folds over like a Pac-Man and it binds to proteins. And what happens? These two particular amino acids are very close to each other in the active solution shape. And so for a long time, there was a debate about whether what this protein structure really was, or was the experiment wrong, because they were debating about the crystal structure versus the actual live structure. And so now, we know there's a lot of flexibility in the point in the middle. In the crystal structure, both the lobes in this position or that position are very, very similar to the crystal structure. It's just that hinge point can move, and you didn't see that in the crystal structure. Another way that we tend to look at this and try to find three-dimensional structure is using NMR. NMR is quite a powerful tool, okay? And we know NMR because you guys learned the single-dimensional NMRs, right? And so you get peaks, right? And if a peak is a triplet, that meant what happened to its neighbor? If something gets split into threes, how many neighbors did it have?
Two.
Okay, and so you can do that kind of thing. But here's the problem. There's so many protons in a protein. We don't look at it in the single way that you do. The peaks would be all over the place. You wouldn't be able to distinguish anything. So we have to use some more powerful techniques, okay, and derive some data from it in a different way. We're going to use what's called the nuclear Overhauser effect, the NOE type of experiment. It is a, instead of a single, we're going to do a two-dimensional analysis, okay? And so what happens here is you're going to get interactions between nuclei, not directly connected, but if they're in close proximity, within less than 5 angstroms. So the NMR that you know about, they have to be right next to each other, connected. Here, if they're locally in space very close, you're going to get a signaling. Okay? And so what you're going to do is you're going to have that normal NMR data in one direction, and I'm going to plot the normal NMR data in the other direction. And what I'm going to do this, if there's any interaction between the atoms, I'm going to get a signal. Now here's the ones that are not important. One interacting with one. Okay, that's not important. That's basically a one-dimensional analysis. This is what happens. if I get a signal between 2 and 5, what will happen here? Here's 5 and there is 2. If I'm getting a signal meaning that they're close, 2 and 5, I'll get a signal off the diagonal. And so I'm going to look for everything that is off the diagonal and go back to see what two things were interacting very close to each other. Okay? What will happen is this. You will get all of the signals off the diagonal and you'll say, this one had to be connected to that one, and that one had to be connected to, well, really you get it this way. This has to be connected all the way over here. And that one has to be connected all the way over here. And that one and that one and that one and that one are connected. And this one and this one and this one are connected. And so you know which ones connect. You also know the Ramachandran plot, what angles you can have. And what you do is you back calculate. How in the world, what structures could you get to have all of those interactions making at the same time? And what you do is you calculate 10 to 20 different types of structures that would fit all the data that you have. And you can see that in many of these cases, all 20 of them have an overlap of what that fold should be. Now, there are places in which, check that out. Is there any consistency here? No, this is where we call random coil. We're not exactly sure where it is. So what will be the refined and what we report is we're pretty confident about these ones right here. There's some beta sheet, there's some alpha helix, and then we just have this coil here, which is probably floppy. There's not a lot of interaction between the proteins, so it could have a lot of movement, and we calculate there's a lot of movement there, and so we can report this. All right. Attributes to this. Usually a better structure in terms of more closely aligned to what is real. But still, the concentrations that you have to get to do this in the experiment is higher than in the cell, so that might perturb what the structure should be. Also, there's a limitation on how large the protein can be. At the time we did the notes, it was about 30,000 kilodaltons, which is smaller than most proteins. We're moving it up. Our more powerful NMRs actually probably has moved that up a bit, but we're still limited to 40 or 50,000 in terms of the largest piece. So sometimes we can't study the whole protein, but we could study a motif. We could clip out a motif and look at that fold. It's limited. Okay. Now, all of the different types of information that you can get, X-ray, NMR, lead to a collection of known structures, okay, of which they need to start to agree. If they don't agree, then you have more issues and you get lots of debates. But this is what can help us out. As we collect structures and we know structures, we also know their primary sequences. And we know their patterns of primary sequences. And if we see the pattern of the hydrophobic in the primary sequence of something that is known and it matches up with something that is unknown, we have a pretty good guess that that protein may fold in the same way. And so as we get more and more data, we are able to guess, theorize, make a theory of what a protein might be based upon all of the data that we know. All right. For some reason, we shift into some laboratory uses of proteins. Okay, I have antibodies in our notes in a segment of if I can get to it or not. Okay, because I think antibody structure and function and a little bit of immunity really works with the protein function. So it's at the end of the next chapter. If we can get to it, we can get to it. If we don't, we don't. This book only spends a very small portion of it because we use antibodies in the laboratory. So these are proteins that we're going to hijack. They're immunity proteins, but we're going to use them in the laboratory for analysis. Now here is why we use antibodies. They bind very, very tightly, very, very specifically. Okay. So if by chance I have a mixture of all sorts of different proteins in a sample, I could use an antibody to identify, is the protein that you're looking for there? Because you purchase a protein, an antibody that will bind to your protein and only your protein in an entire mix. So it's a way to identify what you're looking at. And so the normal process for antibodies is to bind to antigens. An antigen is something that will interact with antibodies, and it binds at a particular place called an epitope. So the antibody binds specifically in an epitope of an antigen. Okay? An antibody is usually drawn like a Y, okay? Because this is what the protein structure actually looks like. Okay? It's kind of like this. Okay? And the antibody is going to bind, and this is where it binds, where its hands are. Okay? These are the places where it binds. That is right here, there's a binding pocket, and right here where the specificity is. All right? This on the back end, my legs, would be, I'm going to bind to something, and whatever my legs are will signal what happens to that. We do a lot of things with what we bind to. Usually we're trying to destroy them. Okay? Depending upon what it is, we will destroy it in different ways. And we may get to that. Or you can take immunology and really spend a whole semester on it. Okay? Looking at this protein, what do you see unique about this particular protein? Look at the structure. All what? All beta sheet. Okay. Also, it is a protein that is made up of 4 peptides. One long. So 4 bundles all connect together. One short. Called heavy chains and light chains. The longer it is, the heavier it is, the lighter it is, the shorter it is, the lighter it is. These yellow bonds right here, disulfide bonds that hold them together. Okay. And so we're going to use these as tools, molecular tools, because we can create or buy an antibody that is specific for whatever you're looking for. Okay? Now, if you can't buy it, you got to make it. And unfortunately, the easiest way to do that is to use something that has a system to make antibodies, and that are biological systems, animals. Okay? And so, a rabbit, a pig, a horse, you could use just about anything. I guess an elephant would work, but they're kind of hard to keep in the lab, you know. So, but we're going to make antibodies. And so what you do is you inject whatever you want to create an antibody inside the animal. Antibodies are upregulated to detect things that are not supposed to be there, non-self. So if you inject something into the animal that's non-self, it's going to create an antibody against it, or its immunity will start eliciting a response to it. Okay? Yes?
Are there any animals that have been found to be more efficient?
More efficient? Rabbits are used quite often. And these are not the nice little bunny rabbits you see. They get mean. But then let me describe what happens to them and you'll know why they kind of get mean. But rabbits are really kind of a standard, industry standard, because of their the way that they produce antibodies. And so sometimes if you inject something, it will actually be in the circulatory system and not be filtered out very quickly. Those are good antibody producing molecules, but sometimes your molecule is too small and gets filtered out before antibiotic resistance can start. So sometimes we have to take whatever you want and hook it onto something so it stays in the blood system long enough. Okay, so we introduce it to the rabbit. Rabbit's immune system says, hey, wait, this is not supposed to be here. And it starts to create antibodies. Initially, you create a certain class of antibodies called IgM, not important at this moment, but it's one kind of antibody. Okay? One kind of antibody. The normal antibody called IgG, that's the most persistent antibody and most common, doesn't come up for a couple weeks. And so if it's challenged, it'll have an amount of IgGs being produced, and that's the one that you really want over a period of time. And then a booster is given. That means you inject them again with it, and your IgG levels go up. All right. This is exactly what happens when you vaccinate. Okay? You make your body create the proteins and the cells necessary to have enough antibodies in your bloodstream so that if you're exposed to something, The antibodies bind to it and take care of it, get rid of it, so you don't get sick. In this case, we're not making the rabbits, we're not immunizing them for immunity. We're actually isolating. We then drain the rabbit's blood, okay, usually through the ear. That's just not nice because that's where the antibodies live, right? And so we take some of their blood. Now, at this point in time, you don't take all of their blood because once the rabbit starts producing, and it's good for you, it's an antibody that you want to use, you feed the rabbit, you take care of the rabbit, you bleed it every so often, it's just like you donating blood. They're donating blood for laboratory use, and throughout its lifetime, now you have a series. Now, I know PETA doesn't like it, but this is what happens. All right? And so now you've got a mixture in the blood of all different antibodies. You can spin that down, and you can get the red blood cells out, and you're left with the serum. Okay? Really, it's called serum or anti-serum because it has a very large collection of antibodies, but it has every single antibody that the rabbit's making. Okay? That's usually not what you want. You want your antibody. So you do an affinity chromatography where instead of injecting it into the bloodstream, you take whatever your molecule is, hook it onto a bead, you pour all the antibodies on top, all the antibodies that doesn't bind will wash out, and then you elute your antibody. And then you package it up and you sell it to somebody, or you use it. All right? And now you have a collection of antibodies that will bind to your antigen that bind at several epitopes. Okay? That means you have a collection of what are called polyclonal antibodies. The antibody will not just bind one place, but there are lots of antibodies that bind to your antigen on multiple different places on whatever it is. And that's usually okay for everybody, because what you are going to use it for is my protein there? You throw your antibody on there, does it bind or does it not bind? And really, I want it to bind if it's there. Okay, so polyclonal antibody is a wonderful, and it's easy, and it's the first one that we make. All right, but that's not all we're going to use antibodies for. We're going to use antibodies for lots and lots of other things. All right, I don't think I have very many